This lecture is split into two parts, all containing cell structure and organelles. This is material from chapter 4 of your book. So we're going to discuss major differences and similarities between bacteria and archaea and eukaryotic cells, reasons that cells are small, and the structure and function of major organelles. First, we're going to go through where did cells come from. It's generally considered that there are four main ways that cells would have emerged. So four main things that really need to happen for a cell to come into being. The first is that abiotic synthesis of organic compounds had to happen. So things like your amino acids and nucleotides had to come from somewhere, and there weren't proteins, enzymes, a cell to make those yet. So that's the abiotic part. Something that's not living, not a cell, had to make these. So I'm not really big on having you guys memorize dates, but clearly 1953, a lot of stuff we've gone over has happened in 1953. Clearly an important year for science. In that year, Miller, he did an experiment with this apparatus over here on the right. You don't need to remember all the details in this diagram, but it's something for you to look at and consider how he went about this. He did an experiment with simulating the atmosphere of the Earth at that time. So over here, he simulated the atmosphere with the gases that they thought might be there. Now we don't know for sure if those are the exact gases and the exact concentrations that were there, but he had to start somewhere, and they have done several different experiments with different results using different gases here. So what he did was put an electrical current, so something like that might be simulating lightning, through this chamber of gases, and then they waited to see if something collected down here, so something condensed, something was made by those gases and condensed down there in the trap. And what he found with this particular scenario is that there was glycine and alanine. So you should remember that those are amino acids, and he found that those were made simply by having those gases and electricity in the system. Now again, we don't know if this is how it happened, but it's one way that they showed that this is a possibility that you could actually make abiotic compounds if you just had some electricity and some gas. The second criteria is abiotic polymerization. So now that we've got these compounds, now that we've got glycine and alanine, we have to have a way to put those together without having, say, a ribosome to do it. The third criteria is emergence of macromolecules for genetic information. There's got to be a way for us to pass on this information from one organism to another. And it's generally considered that RNA probably came first before DNA. And that's because DNA actually needs enzymes to make it. It can't really come about without those. On the other hand, RNA can actually fold on itself and actually act as an enzyme itself. So they generally consider RNA probably came first. And the fourth component is encapsulation in simple membranes. So all of our cells that we know of have some sort of membrane on the outside. So something had to encapsulate all of these things to make it into a cell type form. Why are cells so small? We have a very defined range from 0.3 microns for the smallest bacteria to 100 microns for the largest eukaryotic cells. Those are diameters. But this is still a pretty tight range. Why don't we have cells that are as big as a human or as big as a small animal? There are three reasons for this. The first is surface area to volume ratio. So surface area, if we look over here, this cube is simulating our cell. Surface area meaning the different sides around that cell. And there are exchanges between the cell and the environment that are going on constantly on this surface area. Then we have to consider the volume as well, so everything inside of this cell. How many nutrients do we need to keep it going? How much waste are we producing, and how is that able to get out of that cell? Now if we have this cell, which is 20 microns on all sides, then we have a total volume of 8,000 microns cubed and a total surface area of 2,400 microns squared. But if we divide that into 8 smaller cells with 10 microns on each side, Now we've got all this surface area in between that we didn't have before. So we've got the same volume, but we've increased our surface area by 2 times what it was. So interactions between the cell and the environment can happen with a lot more frequency than they could in this one big cell. Another thing to consider is diffusion rate. So how rapidly can molecules diffuse through the cytoplasm? If we have a molecule that's coming in up here, say it's some sort of sugar, some sort of nutrient, and it's got to get through down to the middle of this cell, through this cytoplasm, how rapidly can it do that? Versus if we're a smaller cell, it's only got to go this far. So it's cutting its diffusion time in half if it's a smaller area that it has to go. And the third criteria is adequate concentrations of reactants and catalysts. So if we have an enzyme over here and we have some substrate that it has to degrade over here, we have to wait for those to actually interact. They could bounce around in the cell for quite some time before they come in contact, and that reaction can actually happen. If we're in a smaller space though, they're more contained, they don't bounce around as much before they actually come in contact, and then even more so in these small cells, they're very close together. Now we can overcome that in this bigger cell if we compartmentalize those enzymes and substrates. So in an organelle, such as a mitochondria, if we contain all the enzymes we need to make energy and all the things that we need to do that, then those are all very close together already, and we don't have to worry about them coming into contact in this big volume of this larger cell. So keeping the cells in this range helps maximize the good components of all these three. We have 2 overall types of cells we're going to talk about, prokaryotic and eukaryotic. The main difference between those two, prokaryotic does not have a membrane-bound nucleus, and eukaryotic does. There are other differences we'll discuss as well, but that's the main one that was first used. That's not a great system to use anymore because that kind of assumes that all eukaryotic cells are going to be similar, and that's just not true. So while they all have a membrane-bound nucleus, they are not all that similar. Nevertheless, we still use that criteria as kind of our large classification. Now within that, we have bacteria and archaea are 2 groups of prokaryotic cells. So bacteria being single-celled organisms, archaea also single-celled organisms. Neither one of these have a membrane-bound nucleus. You will see, however, that when we're traveling along these lines, and these lines represent evolution from an ancestral cell, that archaea is coming off of the line that leads to eukarya and not bacteria, meaning that it's closer, more closely related to eukarya. So there are actually characteristics of archaea that are similar to eukarya and some that are similar to bacteria. So they found that actually DNA-wise, they're more similar to eukarya, so we're going to classify them that way. Most of our examples are going to be of bacteria and eukarya. Occasionally we'll have one of archaea, but we're not going to touch that one quite as much. Differences between cell types. There are 6 that we're going to go through. The first is the obvious membrane-bound nucleus. Here in our prokaryotic bacterial cell, the nucleoid, so that's just their word for the DNA, it's in this clump here in the middle but not bound by a membrane, versus our eukaryotic cell, which is bound by a membrane here with all the DNA contained. The second one is organelles. So these membrane-bound partitions that have a specific function. For example, your mitochondria or your lysosome. These are bound by membranes. They're containing certain enzymes that do certain functions. Now there are exceptions of bacteria having something that resembles a organelle, and one would be this bacteria. This is Caulobacter, and it puts out this stalk here that is able to gather nutrients, and it actually attaches to things. So there's some kind of glue here on the end that attaches to things. This is kind of separated from the rest of the cell, so it's kind of similar to an organelle, but not quite the same thing. So that's some exceptions. The third criteria, exocytosis and endocytosis. This is exchange of material between compartments. So clearly up here, if you don't really have compartments, that's not going to happen quite as much as something down here where you can send material, say, from your endoplasmic reticulum to your mitochondria. Fourth is organization of the DNA. Bacterial DNA is circular. It's one big piece in a circle. And this shows you it's kind of wound around itself here, but one big piece in a circle, versus our cells, which have several separate chromosomes. They're linear, so one piece that has a beginning and an end, and they're compartmentalized with these proteins called histones, so they're wound around it in a very organized structure. There are also something called plasmids in bacteria. We're not going to go through that now, but that'll be coming up in our lab here in a couple of weeks, where we'll talk about that. The fifth criteria is segregation of DNA. So this bacteria cell, when it's ready to make a new bacteria cell, it's going to replicate everything, including its DNA, and then it's just going to divide right here down the middle. That's called binary fission. Versus mitosis, which is the process that our eukaryotic cells go through, which is a very specific way of partitioning the DNA into two different daughter cells. And our sixth criteria is the expression of the DNA. So in prokaryotic cells, you can have one mRNA, which can then code for several proteins. We call that an operon. In eukaryotic cells, in contrast, we have one mRNA, usually just codes for one protein. There are exceptions, but that's the general rule. So you may want to go through and compare the eukaryotic cells that we're going to be talking about mostly. This is an animal cell here on the left, and this is a plant cell here on the right. So things that you may notice that are very distinctive between the two. Plant cells have this big, thick cell wall around the outside that gives a lot of structure and rigidity. So the plants you see, they're not very fluid, they're not very squishy. They have a rigid structure to how they look, and that's in part due to this cell wall that's surrounding it. Another thing you might notice are the chloroplasts. These are involved in making sugars and making oxygen. These are not found in animal cells, only found in plant cells, and that's what gives the plant cells their green color in leaves. And a third difference you might see is this big vacuole. So in plant cells, you usually have these big vacuoles, usually filled with a watery-like substance, and sometimes there will be nutrients and pigments in those. You don't have those in animal cells. In plant cells, this is mostly going to provide structure. Again, if you see a plant that is wilted, it's because it's lost some of the water in this vacuole, and it's losing some of the structure in its cells. So that's the end of this video, and go on to the next video to talk a little bit more about our specific organelles.